Good morning. Today we are going to see what is erythropoiesis. So before going to learn about erythropoiesis, we should know what are all red blood cells. So and functions of red blood cells. Red blood cells. This is also called as erythrocytes. These are biconcave disc shape. As you can see, it is a disc shape, and in center we can see it's very thin. So it is about 7 to 8 micrometer in diameter. 7 to 8 micrometer in diameter. A single drop of blood contains millions of erythrocytes, and you can see thousands of leukocytes in a single drop. एक single drop में millions of erythrocytes यानी कि RBC और WBC, thousands of WBC we can see in a single drop of blood. So specifically if we see, males have 5.4 million erythrocyte per microliter and females have 4.8 millions per microliter. And these RBC, red blood cells, will be surviving up to 120 days within the body itself. Suppose if we take out of the body for any kind of diagnostic test, if we keep in a test tube, this RBC lifespan will be 3 to 4 weeks. 3 to 4 weeks. So next we'll see functions of red blood cells. So what are all these functions of red blood cell? The main important primary function is transportation of oxygen and carbon dioxide. So here RBC will pick up the inhaled oxygen from the lungs and transport it to the body tissues. ऑक्सीजन इनहेल करने के बाद में ये आरबीसी या एरिथ्रोसाइट ये ऑक्सीजन को लंग्स से इट विल ट्रांसपोर्ट टू द बॉडी टिश्यू को ले जाता है ऑक्सीजन, एंड ऑल्सो इट विल पिकअप 24% कार्बन डाइऑक्साइड फ्रॉम द टिश्यूज टू द लंग्स. पहले लंग्स से टिश्यूज को, फिर 24% टिश्यूज से लंग्स को कार्बन डाइऑक्साइड. सो हियर गैस एक्सचेंजिंग ऑक्सीजन एंड कार्बन डाइऑक्साइड. Next, it will maintain acid-base balance and also contribute to the viscosity of blood. It will regulate the viscosity of blood. Viscosity, the meaning of viscosity, that is the state of being thick, sticky, semi-fluid. अगरचे blood बहुत thick है या बहुत fluid में है, fluidity ज़्यादा है, बहुत क्या बोलते हैं उसको, thin. So there might be a disorders. So it will maintain blood consistency. So that is the meaning of viscosity of blood. It will regulate the viscosity of blood. And other one is influence the erythrocyte sedimentation rate. What is this erythrocyte sedimentation rate? So erythrocyte sedimentation rate is a test where RBCs will settle down in the bottom of test tube. If we collect some blood from the human and if we put into the test tube, क्या होता है? RBC will be settling down. So there is a specific time, but lot of time will be taking for settling RBC into the test tube, settling down. If the rate is fast, मतलब RBC ज़्यादा जल्दी नीचे आके रुक गए, settling down, that means there is an indication of inflammation. So that's why in specific diseases we'll check the erythrocyte sedimentation rate. So these are the functions of RBC. First one is a transport of oxygen and carbon dioxide, and it will maintain acid-base balance and contribute viscosity of blood, influence erythrocyte sedimentation rate. So now we'll see erythropoiesis. So what is this erythropoiesis? The process of manufacturing of RBC, red blood cells. So these red blood cells will go through several stages to form a matured erythrocyte. So here you can see RBC production will occur in a bone marrow. These RBC ये red blood cells कहाँ पर पैदा होते हैं? Where it will take birth? Mainly in a bone marrow, skull, ribs, vertebrae and in the ends of long bone. So this RBC production will takes in the bone marrow, skull, ribs, vertebrae and in the ends of the long bone. So there are certain stages. So here first stem cell. So production I said that it will take in bone marrow. First stage, that is hemocytoblast. So hemocytoblast, at the earliest stage you can see the cell size is large and has no hemoglobin. Remember, in a first stage, that is hemocytoblast, the cell will be large and there is no hemoglobin. No hemoglobin. Next, the size of this is 20 micrometer. 20 micrometer in diameter. So the size of cell will be 20 micrometer in diameter. So first stage is hemocytoblast. The cell will be quite large, that is about 20 micrometer in diameter and has no hemoglobin. Next, committed cell are called as pro-erythroblast. In that we can see early erythroblast and late erythroblast. Early and late erythroblast. So what happened in a pro-erythroblast? A little later, size will be reduced. So here it's a large, like 20 micrometer. So here when it come to the pro-erythroblast, the size will be 14 to 19 micrometer. So here in a pro-erythroblast, it will acquire hemoglobin. So in early erythroblast, what happened? Ribosome synthesis will occur. Then here hemoglobin accumulation will takes place in this pro-erythroblast and still has a nucleus. You can see this is a nucleus inside. So फर्स्ट इज द हीमोसाइटोब्लास्ट, नेक्स्ट इज द प्रो एरिथ्रोब्लास्ट. इसमें दो स्टेजेस है, अर्ली एरिथ्रोब्लास्ट, लेट एरिथ्रोब्लास्ट. अर्ली में क्या होता है? राइबोसोम सिंथेसिस होता है. देन फेज 2 में हीमोग्लोबिन एक्यूलेशन एंड स्टिल इट हैज़ ए न्यूक्लियस. सो नेक्स्ट normoblast. So here in normoblast, the next stage is size will be like matured cell only, but it still possesses a nucleus. So when there is a ejection of nucleus occur, that will be termed as reticulocytes. So now this reticulocyte is up to 8 to 9 micrometer. So this reticulocytes, final maturation stage is reticulocyte. So here in the normoblast what happens, still it possesses a nucleus. When the nucleus is expelling out, it will be converted into reticulocytes. So here reticulocyte size will be 8 to 9 micrometer in diameter. So this all process, this all till reticulocyte, this process will occur in bone marrow itself. So after reticulocyte formation, these reticulocytes will be discharged into the bloodstream. After few hours, what happened? These reticulocytes will leave their reticular pattern and will become mature red cells, erythrocytes. So this is the process of erythropoiesis. First, is hemocytoblast, where the size will be 20 micrometer. Then it goes to the next stage, pro-erythroblast. In this, early erythroblast you can see and late erythroblast. In early, ribosome synthesis will occur, then it will acquire hemoglobin. Then after next stage, it will convert into the normoblast, where ejection of nucleus takes place. It will be termed as reticulocyte, converted into reticulocytes. So this whole process will occur in the bone marrow itself. So then the reticulocytes will be entering into the bloodstream and will become matured erythrocytes. So this is about the process of erythrocytes. If you have any kinds of doubts, you can comment and contact me. Thank you. We have seen what is RBC, functions of RBC, and erythropoiesis, process of erythropoiesis. Thank you.